Next up, we have the Medical Informatics and Systems Division.
Hi, I'm Skip Garner. I direct the Medical Informatics and Systems Division. The way in which we're going to do this today is that each of the principal investigators, is providing one slide, and it's just to kind of stimulate the general description of the kinds of work they do. And so I also want to emphasize that we're really into translation and to getting our results into the clinic. And as such, we not only have our science in here, but I think, last count, at least three spin-out companies for the division. And I want to start this off by actually giving you my slide before we move on to each person. And so what this is a description of our work where we now have developed techniques to rebuild every genome, especially the junk part of the DNA that's often ignored. And we focus on repetitive elements or microsatellites or sequences that go like CAG, CAG, CAG. What we found when we started to do this process is that all current assembly engines get it right about 20% of the time. So only 20% of the time do they obey Mendelian inheritance, which is something that we kind of believe in. And so we rebuilt all those algorithms and went to an entirely different approach, and now we're building all the loci at microsatellite at about a 96% accuracy, which then allows you to do discovery, which was not possible before. So this is one example of the platform technology. Now we've done about 10,000 genomes, completely rebuilt them from scratch for about a dozen different cancers. This happens to be breast cancer. Through the analysis of about 600 or so germline sequences and a couple hundred matched genomes, we were able to identify 55 loci that were robust and independently determined and passed stringent false discovery rate tests, et cetera. And once we identified those, we could do a number of things with them, including building a signature that could predict the potential for risk for developing breast cancer from looking at the germline, i.e. from blood. And this just happens to be the results. The blue bars here is the general population, the matched population there of people who are cancer-free. And they primarily do not have the alleles that contribute to the risk. And these are 55 new loci with all new alleles that were previously undiscovered. However, when you look at the germlines of the breast cancer patients, not the tumors, but the germlines, you can see that they kind of sequester in an area where they have genotypes that are unique to cancer. And of course, you can see that the red is the germline, the orange is the tumor. The tumors also have inherited this pattern of risk markers. And what this has allowed us to do is to identify using receiver operator curves to draw a line and then to develop a diagnostic test where if you're on this side diagnostic test, you're at low risk for developing cancer. Here you're at high risk. So we've done this for prostate cancer, a variety of brain cancers, ovarian cancer, et cetera, and published all this stuff. So this allows us to now move this to the clinic, and we're now independently validating these at other universities across the United States, across the world, sorry. And for our test, the sensitivity is about 88%, the specificity is 77%. Now, the reason why I want to bring that up is that breast cancer is the best genomically diagnosed cancer in the world, and it's largely through the analysis of the BRCA1 and 2 genes that, as you know, if you read the papers, Myriad and others have kits for that. And just so you know, about 8% of the people who develop breast cancer are positive for that test, so their sensitivity is around 8% if you measure it in a similar way. The other thing that you should know is that 12% of all females will develop breast cancer at some time in their life, and so as a consequence, the specificity at best you could get is something at the 85 to 90% range. Okay, and so what this is one of the many new diagnostic tests, the basic science that underlies the new diagnostic tests that are now impaneled so that we can do all this stuff. And as opposed to having one test for each gene that runs you $2,000 or $3,000, Our cost of goods currently is about $200 to test your risk and also do a variety of other diagnostic tests to partition you for all these at one time. Okay, so thank you very much, and next slide, please.
Okay, I'm excited to tell you about our research in the lab of integrative genome analysis. We compare multiple genomes from a range of species, basically ranging from like fruit flies to whales. We also, in collaboration with Joseph Besagania, we analyze transcriptomes in relation to infectious disease. But today, I wanted to tell you about our recent discovery of a very interesting and intriguing phenomenon related to ribosomal RNA and ribosomal DNA, using Xenopus frogs, African cloud frogs, as a model system. Why frogs? It just happens that they're very easy to breed, and turns out that we can hybridize very distant species. The time of divergence between the species that we hybridize in the lab is like 70, 80 million years, which is quite amazing. So when you want to compare it to like primates and humans, that would be humans on one end and gibbons on the other range. So what it means is that they preserve a very old, very distinct genetic markers that we can use, which is very important for ribosomal RNA and DNA, because these sequences tend to be very conserved. And I can't really emphasize how important part of the genome it is. Maybe I'm reluctant to say it's the most important part of the genome, but it's definitely the most ancient part of the genome, because it probably derives from an environment where there were no cells, and probably there was no DNA either. It's an environment called hypothetical world called RNA world. Also, ribosomal RNA is the main structural component of ribosomes that are very important for metabolism and a basis of translation. So very fundamental feature of life. And what we discovered was using interspecies crosses between Xenopus species is genomic imprinting of paternal genes. So right after fertilization, paternal ribosomal RNA genes get silenced in the egg. And we don't really understand why that happens. We speculate that this is related to genomic conflicts, very ancient features of genomes. And probably this epigenetic reprogramming is sort of like epigenetic immune response to these conflicts. And this is something that affects at least 50% of all transcriptional output in the cell, which is quite significant, because this is what most of RNAs in the cell are. This is ribosomal RNA. Somehow it was completely overlooked because there were no markers for these sequences. We have the markers because of the divergence between the species. So this is a very significant epigenetic reprogramming at the transcriptome level, but also it affects a significant part of the genome, because ribosomal RNA genes are organized into multi-copy gene clusters, and these clusters form a structure called nucleolus that is the main production center for ribosomal RNA. And it's actually a distinct feature of nucleus that can be seen under a microscope. So we discovered that looking at actually another very interesting feature of Xenopus genetics, which is a nucleolus or nucleolar dominance. So when two parental species are hybridized, they both have two nucleoli per cell. The DF1 hybrid has only one. So it was speculated before that it must be related to the pattern of expression of ribosomal RNA, but again, no one really expected parental imprinting or genomic imprinting in the system. So just to sum up, this is a new discovery of genomic imprinting in a very important part of the genome that affects at least 50% of transcriptional output in the cell. And it's probably related to genetic conflicts and sort of epigenetic responses to these conflicts. Thank you.
So it's been exciting to visit and see friends and collaborators. So I'm excited to be back for a visit. I apologize for the brain dump. I'll just talk about a couple things that have been interesting from our group. There's been a lot of promise, and people have spoken of that promise for personalized medicine, precision medicine. We heard about it at the State of the Union. And the technology exists and is being used every day by us, by folks at other universities. But there's a barrier to taking genomics from where it is here and bringing out to the clinic, to the bedside, even to kind of high throughput diagnostic labs, like for example Myriad. And so I've been very interested in the discussion of how to standardize and how to benchmark and understand the performance of sequencing, of how we do genetic analysis, both so that we can standardize it across collaborating groups. And everyone here obviously is on board with the idea of team science to drive really big discovery in medicine and in genetics. And also figuring out how to lower the barrier for entry for others to join into the space. And so just a couple things that we've been working on. One that I like to highlight, I have, much like Skip does, I have a very strong fascination with short tandem repeats for a whole number of reasons. And they are very hard to characterize from genomes. And so my group has done a lot of work trying to figure out the best way, not just to identify and genotype STRs in various genomes, but also how to set benchmarks and performance metrics to know that we're actually generating data that's suitable to pull that kind of information from. And so we've had a very lengthy and I would say very profitable and rewarding collaboration with the 1,000 Genomes Project, where we convinced the 1,000 Genomes Project, they're about to publish their last paper. They're going to include STR data, which for me is a thrill. It's been small indels and SNPs to date. There's a paper that I'm highlighting here. It was published in Genome Research in collaboration with Jana Erwich's group. That actually is an exhaustive catalog of all the STRs we were able to find in the 1,000 Genomes data. And this data is already being used now in Genome in a Bottle, in the Global Alliance, which you guys may have heard of, which is like the new Meta 1,000 Genomes project that's even more international. And so I'm very excited to have participated in that. Another trajectory is kind of coming back to lowering the barrier of entry so everyone can get into genomics. You buy that MySeq that's finally affordable if you have a big budget. And the data comes off the machine, and what do you do with it? And so we've built some tools. I couldn't find an exciting screenshot to share with you guys, but if you search on Google for GCAT, G-C-A-T, you'll find it. It's a free online platform that we built. It allows you to download raw Illumina data and Ion Torrent data, but our focus is on Illumina. And you can take it, run it using your lab's secret tools, run it using open source tools, download a free trial of some company X's tools, and run variant calling or mapping through that data. And then you can upload the results right back to the website, and you get an instant report that shows you how you've performed against a number of metrics that, for example, look to see that you're capturing the full spectrum of indels, not just insertions or deletions, SNPs that obey the kind of TI-TV ratio you'd expect, and also a gold standard kind of list of calls that we've curated as part of the Genome in a Bottle project with 14 different, I think, sequencing runs across several platforms and universities across the world. And so, and that's this most bottom paper right here. This was the call set that we built. And so what this allows you to do is kind of see where you perform, or the tools that you're using where they perform. And it gives you a starting point to then decide how to optimize or how to build new tools that'll be able to further improve, the specificity or accuracy or sensitivity of what you're doing. And it's an interesting problem because there probably isn't one set of tools or one set of metrics that will be the final way that you do sequencing. If you're involved in carrier screening, which I think is going to be the next big kind of killer app of genomics for the public, then you don't really care about false positive rate, but in fact you're extremely concerned about sensitivity. You don't want to miss that one variant that might underlie a disorder that you might pass on to your child. On the other hand, if you're doing something kind of in the cancer space, and you're looking to identify what pathway was mutated, then false positive rate's really important. If you highlight all the pathways, then you haven't really provided any value for the physician. So yeah, I guess to summarize, we've been very excited to build these tools, not just build them, but make them open and accessible for anyone. We welcome industry folks, academic folks, citizen scientists, everyone in between to use it. The data can be reused for any purpose you want. In fact, there's a couple of folks that have now taken we allow you to share the benchmarks and the reports off GCAT. You can generate images for your papers. You can export it to like blog posts and social media. And there are a number of companies now that are using these figures in their white papers. There are, believe it or not, academics that have put these figures into their manuscripts. And so it's exciting because it's not just providing a way for us to all discuss, but it's actually being utilized and hopefully benefiting folks. So thanks.
Okay, so I'm from Jean Picou's lab, and I'm going to represent the lab because Jean's out of town right now. One half of the lab has traditionally been more involved in synthetic biology and the development of tools for synthetic biology, but a lot of that work has now moved over to a startup company that Jean has started. So what I'm going to talk about is our work on the study of cell cycle regulation, and we use the budding yeast as a model system to study this. Our approach is to take a modeling experiment cycle approach to study this. And in terms of the modeling, we do a lot of that in collaboration with John Tyson's lab over in the biology department. And the approach is to use models to try to understand the molecular dynamics underlying cell cycle transitions and develop these very complicated models to help explain what's known about the molecular dynamics, and then use the experiments to fill in gaps about the molecular dynamics using molecular and synthetic biology approaches to get quantitative data that we can then put into the model and parameterize it. And then what we do is test predictions of the models by perturbing these systems, mostly through mutations in the budding yeast, and then looking at the phenotypic outputs of those perturbations and seeing if the model correctly predicts what the outcomes are. And then we feed back into the models to better refine the models and represent the new data that we generate. So the overarching goal of this is basically to understand the network dynamics that lead to irreversible transitions between the different stages of the cell cycle, and also some of the quality control mechanisms, otherwise known as cell cycle checkpoints, that inhibit these transitions when there are problems in the previous phase of the cell cycle. By taking this approach, we hope to better understand the dynamics of cell cycle regulation, and a lot of this will feed into our understanding of cell cycle changes that occurred during embryonic development, tissue homeostasis, and when the cell cycle is perturbed in diseases like cancer. That's it.
So I'm part of Skip's group, but I'm branching off and beginning my own line of research. And so what I want to talk to you today is one of my focuses is on continuing to mine existing genomic data for new information and ways that we're doing that. And so if you'll allow the analogy, I like to think that in the decade or so since genomics has emerged, we've gotten really good at getting what I call a first cold press genome, and that's like all this data exome sequencing. We've gotten the tools and the ability to really extract a lot of great information really fast from genomic data. But one of the overarching hypotheses of my goals is to keep pressing and see what else we can extract after kind of the first extraction is completed. And so one of the ways we're doing that is by looking through what goes into the unmapped reads from genomic and exomic data and saying, can we start piecing together additional information from what's thrown into the trash heap, thrown into this unmapped reads basket. And so there's a lot of different reasons that reads are thrown away into the unmapped read bin, if you will. And a lot of that is low quality from sequencers, but sequencers are getting better and better, and there's actually being high quality reads being discarded into unmapped read no man's land. And really we're being able to then go through this and extract these and piece them together and find that, yes, these are human sequence, we can get them in high quality and start rescuing these things. And we hope to eventually start getting this at enough depth to integrate these back into the human reference and start adding these additional sequences to the human genome. And you'll see that actually, if you look here, when we've done this, and we're focusing on microsatellites, just because that's the tools that we've developed right now, and there's a lot of information here. But if you look at the microsatellites that we've looked at in these unmapped reads, we can see that if you look at the red bars, these are the ones that are kind of known in the current reference genome. And when we look at the unmapped read category, we start seeing that there's a lot of 5-mer microsatellites that are just being heaped into this unmapped reads. And so we're starting to be able to analyze this and say, what are all of these? What purpose do they serve? And we think that many of these are centromeric microsatellites, and we can start piecing together these. And so I want to touch real quickly on also another way we're doing it, and it's been recently published, so there's more information out there on a new measure of somatic microsatellite variation, where we're looking beyond the genotype to look at additional alleles that may be present in sequencing data to see if these additional alleles can give us an idea of the amount of variation within an individual's genome. And we're finding that people with known defects in DNA repair mechanisms have a different pattern of somatic microsatellite variation than do healthy cells. And so these two ways, we're kind of extracting more information from genomic data.
Okay, good morning. I'm David. Before I start my work, I would like to say thanks to my group members, in particular Mingyang and Kevin, who did excellent work, so allow me to share my story with you today. We have been interested in brain development for a couple of years. Brain, it looks like your cell phone. So let's imagine everybody here gets a cell phone, new cell phone today. So 1 year after, you will find that every cell phone is different from each other now. You find different pictures, contact information, music, and some games you don't know from internet, right? Brain is the same. When we grow up, now everybody has something different in your mind. So what happens? We found that a protein or a gene, EGR1, is super important for this process. Now let me walk through how we found this protein, why I say this protein is so important. We start with a very simple question. There are so many cells in our brains, right? We know that. Are they the same? The answer probably is quite simple. No, they can be identical to each other. How are they different? So this becomes a huge question to us. And how they become different, right? So these questions took us 3 years to address that. So one way is that, let's do it this way. We break the brains and then take out each cells and sequence each cells. There are 100 billion cells in there, so this way probably doesn't work out well. So we combine an experimental approach, and this was published last year, and a computational approach. So I'm not going to go to detail with you. Just believe me, we can do it. So we combine these two together. Now, we dissect the brain mycelium, and we find that in our brains, around 30,000 genomic loci, they show methylation difference. So what is methylation? So methylation is like on-off switch. When DNA gets methylated, it shuts down the genes. When unmethylated, it turns on the genes. So this on-off switch is very important for brain development. And now here is showing up is these regions, that are important regions. Now we ask, do you think they can work together or they work alone? No, they are actually work together. So work together, it means that you can find a group of these switches. When they turn on, they all turn on together. When they turn off, they all turn off together. So we group them, these together. And then we ask the next question. If you on-off switch can work together, who control you? Then we find the protein, EGR1. So EGR1 have tons of binding sites on our genome, on these critical switches. So what is EGR1 then? EGR1 is a neural activity-induced transcription factor. So it's too complex for you, then just think it can bind to DNA and recruit other protein to change the switches. Okay, so this is a process we develop, but this process can apply to any other complex cell system. So imagine you have a tissue, you want to know what's going on within that tissue. So we can use a similar approach. But for my talk, take-home message is that if you give your friend a hug today, the protein EGR1 can help your friend to remember this hug for a long time and the level of DNA methylation. Okay, thank you.
Thank you so much.
Any questions?
Yes, what's the evidence
Yeah.
that EGR1 can really do that? This is all correlation, so where's the causal evidence?
So this is predict, right? EGR1 is predict in the beginning, but we need to provide evidence. This is a very good point. We start with a lot of computational work. and in the end, we need to prove on the bench. So I trust bench. We do the chip sequencing. Basically, you using antibody pull down this protein, whatever DNA bind, and we check the methylation change through the development. And you see that this methylation change only occurring in neurons, and during the development, it gradually lost the methylation. So that is how it happened. And this protein is very well known. People know that for many years. You can find 1,000 papers published for this protein, but they just don't know it changed your methylation. Okay? Does that make sense? I can show you more data.
Actually, you just don't have to just believe him because he says to believe him. You can read his papers.
Oh, yeah. And the paper is pretty much done. Yeah, I finished the paper. Yeah, thank you.
Any other questions?
Hey, David. I have another question. So I know that some of your work has to do with epigenetics in early development, and I was wondering whether you can, I'm very interested in how this, environmental factors might influence at the epigenetic level. So how early these changes happen?
Okay, very good question. So a lot of detail I need to bring it up. EGR1 protein expressed low in the fetal, and then after baby born, it starts to go up. So it's critical for the postnatal early time, early 3 years. It jump up. And now this protein, what it does, that when I talk to you now, you start to think, right? And in millisecond, this gene is go up. And then it go to nuclear, do something there. And then 1 hour, it will drop, go back, calm down, quiet. So this process is really quick. And this happening, I believe, personal belief, is a postnatal life. And the beginning of our life period is so important. So many long-term memory things established at that long time. But I know the marker will switch are, but understanding the switch is going to take me another, I don't know how many years. So, hope answer your question somehow.
Great. So this is a non-EGF question, but for the rest of the panel, because I know the MIS division has a lot of focus in translational diagnostics. And so it's becoming, more and more evident that these algorithms tend to have different benchmarks, right? And so where do you think your algorithms, or where do you think the standardization process is really going to go in the next 5 to 10 years? Because this is something that I think will be heavily regulated because it brings in human health. So.
Well, let me start to answer that. It's not on. The algorithms to date have been built really for a particular purpose, to assemble the genomes when there was no genome 10 years ago, to reassemble genomes on top of it. And really, the kind of efforts has been to do things like, well, let's call the most number of SNPs or most number of variants that we have, as opposed to high quality variants, things that, that are potentially clinically valuable. And so the effort now is really turning to trying to develop technologies and techniques to not just call variants, but to also call robust variants that have some clinical implications, that can be exploited, that are actionable. So you're going to actually see a lot more work in the future to come up with things that are actionable. So that, and I can tell you, if you're actually trying to get anything into the clinic or into an investor to get it into the clinic, the word actionable is an essential part of that. And so I think that that there's going to be less kind of academic freedom to just call as many things as you want, as opposed to calling things that are high quality. David, you probably got comments on this.
Yeah, sure. I have. Sorry. Yeah. Oh, if it's on, I didn't realize. So I have a response. I think right now the whole genomics market, whether you're in the academic or in the industry sector, is very fragmented. It's like in the 90s when people were buying video cards and motherboards and assembling computers, and then those were consolidated and replaced with verticals. So I think if you look, for example, at what the FDA is doing now that it's pushing for LDT regulation, and they're already telling you, you don't have to guess what they're expecting in the next 5 years. And in CLIA and CAP, what you're going to find is that more and more, and this has already happened, like in CLIA and CAP labs that I've been part of, they don't really care what algorithm you're using, and they don't care what sequence you're using. What you're doing is you're offering a test, which is a vertical that integrates the sequencing, whatever you do with the data, and the reporting and annotation and everything else. And then you benchmark that against tools like GCAT, which has actually been cited as a way to do FDA and CLIA and CAP validation, against other truth sets, against the CAP. Like CAP is now going to be sending around kind of like test samples. You run your thing and you see if it matches up. And so I think the algorithm becomes like one tiny piece. It's irrelevant to optimize an algorithm because it's heavily coupled to how you do the sequencing, what platform you use, even the kind of input DNA going in. So I think those details will kind of fade. And then what's really going to matter is just if you advertise that you can pull out this amount of information, that you can pull it out. It's the same problem 23andMe faced and everyone else is facing it. It doesn't matter if you're sequencing on an array or whatever you're doing, they're just going to want to know that your actual full stack of technology produces something that is what you're advertising it produces. So I think that's what's going to happen. You won't see any more companies that are offering bioinformatics service or sequencing. There'll just be people that offer full tests.
Okay, great. Thank you for the thorough answer. One question from Bishop, and then we got a...
Well, I was out for a couple of minutes. I don't know whether anybody asked or not. This is for Mahalik. The question is, you are calling it antagonism, and it's ancient. How far can you go down to see where it started. Without mitochondria,
I guess the question is about genomic conflicts. They're very ancient. It refers mostly to eukaryotic genomes, where you have maternal and paternal genes, and it's especially an application to, it applies to organisms that have like parental care. There's some intimate contact between offspring and mother. So there is there is a conflict between, first of all, mother and offspring, which is quite known, but within the genome also there is a conflict between maternal and paternal genes. And also, you mentioned mitochondria. Yes, mitochondria are a part of the conflict because they come from mother only, so it's maternal inheritance. And there are some described examples of conflict between mitochondrial and nuclear genes. But this one that I described is just apparently it's all within ribosomal DNA, which also the structure of the complex has a hallmark of genetic conflicts because of its repetitive nature. There are multiple copies of the genes, so there is some redundancy that doesn't make any sense. And I think the easiest explanation is that the whole system is driven by some ancient genetic conflicts.
Okay, great. Well, we can give a round of applause to the MIS division for a very nice presentation.